Welcome to Botany A to Z. In today's topic, we'll be discussing the significance of photorespiration. We'll see the demerits of the process, and we'll also discuss the plus points. Photorespiration, as we have already discussed in the earlier video, or you can find the link of that video in the description of this video. In the photorespiration, we have discussed that in C3 plants, when the carbon dioxide concentration or the carbon dioxide partial pressure is low, the enzyme RuBisCO acts as oxygenase, and as a result, by using oxygen, by consuming energy, it breaks down the RuBP into 2-phosphoglycolate and 3-phosphoglycerate. So it is breaking down the already formed sugars with the help of oxygen, hence also called as photosynthetic carbon oxidation cycle. So this all you have seen in the earlier video. You can recap it again by finding the link in the description. Here we'll be discussing the demerits and the merits. We'll be discussing the demerits first because as a whole the process is considered to be a wasteful process. The demerits include loss of carbon dioxide 20 to 25%, consumption of additional ATPs, and decrease in efficiency. The merits include protection against photooxidation and against the reduction, production of hydrogen peroxide, and production of many other byproducts metabolites like amino acids. Let's discuss the demerits first. As you know, as we have discussed in the C2 cycle in the photorespiration video, that in this process the already fixed RuBP is broken down in the chloroplast, and finally 20% of the carbon dioxide is lost. When this cycle reaches the mitochondria phase, 20% of the carbon dioxide is lost when two molecules of glycine combine to form serine. A carbon dioxide molecule is lost. Since the cycle starts from a 5-carbon sugar RuBP, and in the mitochondria we are losing one carbon dioxide molecule, that means 20% of the carbon dioxide is being lost. So we are losing carbon dioxide from the already fixed carbon dioxide. From the already formed RuBP, we are losing carbon dioxide, and that too by consuming ATP. So we are consuming ATP, the process is consuming NADPH. It is energy dependent process, and the energy is being consumed for what? For losing the already fixed carbon dioxide. So this decreases the efficiency of C3 plants. You have seen in the when we discussed the difference between C3 plants and C4 plants, or we discussed the C3 cycle, C4 cycle. In C3 plants, only 18 ATP are used to form one glucose molecule or to fix 6 carbon dioxide atoms in the C3 plants. But in C4 plants, you have to use 18 plus 12, 30 ATP to form one glucose molecule. So although 30 ATP are being used in C4 plants, but still C3 plants are less efficient. C4 plants consume more ATP, but still C3 plants are less efficient. Why? It is all because of the photorespiration. It is all because of the oxygenase activity of RuBisCO. Since 20% of carbon dioxide is being lost, so we will have to use further 20% ATP to regain back that carbon dioxide. Moreover, while losing this carbon dioxide, ATP are being consumed. So for a loss, ATP are being consumed. As a result of these reasons, despite using only 18 ATP to form one glucose in the C3 cycle by the C3 plants are still less efficient just because of the oxygenase activity of the RuBisCO, just because of losing this 20% of the carbon dioxide and consuming additional ATPs for this process. As a result, it is considered as a wasteful process, but there are some merits, there are some plus points as well. Since as I said earlier that when the temperature is high or the carbon dioxide concentration is low, RuBisCO will not be able to act as carboxylase. As a result, the large amount of ATP, the large amount of NADPH that has been already produced in the light reaction part of the photosynthesis, and obviously when the temperature is high, So further more ATPs and NADPH has been produced. Since carbon dioxide concentration is low, since RuBisCO is not acting as carboxylase, that means a large amount of ATP and NADPH will accumulate. As a result, it can cause photolysis of important constituents of the cell, important biomolecules. It can cause photooxidation. Moreover, the NADPH produced in the excess can cause reduction of different important molecules. So in order to protect itself from the photooxidative damage, in order to protect from the photooxidative damage, the excess energy, that is ATP and NADPH that has been produced is being dissipated, by getting consumed in the photorespiration. So when this excess energy, these excess ATP, excess NADPH are used in photorespiration, the plant is that organelle, the cell, the mesophyll cells, the components of the cells are being saved from the photooxidative damage. Moreover, the photorespiration removes many toxic metabolic intermediates. Look, it is actually the decrease in the efficiency of C3 plants is not because of the photorespiration. It is because of the oxygenase activity of the RuBisCO. Photorespiration, in fact, helps us in getting rid of the phosphoglycolate. Phosphoglycolate can be toxic. It can inhibit the triose phosphate isomerase enzymes. So the phosphoglycolate which is being produced by the oxygenase activity of RuBisCO, this phosphoglycolate is converted into other useful constituents like amino acids, and at the same time when the phosphoglycolate is being converted into the useful components, we are saving triose phosphate isomerases from getting inhibited by this. Moreover, in the peroxisome part of photorespiration, H2O2 is produced. Hydrogen peroxide is produced, and this hydrogen peroxide is used by the plants in the abiotic stress responses. It is used to protect the plant from various pathogens. Further, many useful intermediates are produced in this C2 cycle, in the carbon 2 cycle, or the glycolate cycle, or the photorespiration cycle, and preferably the amino acids. In the peroxisome and mitochondria part of the cycle, you have seen that glycine and serine, these important amino acids are produced, and these are then transported into the cytoplasm for the synthesis of methionine. So this is another plus point of the photorespiration. Moreover, the photorespiration connects the metabolic components of the cell and facilitates the transport among organelles such as chloroplast, peroxisome, and mitochondria. So in all, there are demerits of the process, but there are also many useful plus points of the process. We can recall again, the demerits include loss of 20% carbon dioxide, consumption of additional ATPs, decrease in the efficiency of the C3 plants, because the C2 cycle occurs only in the C3 plants, C2 cycle photorespiration. And the merits include protection against the photooxidation and reduction, production of hydrogen peroxide, which is used in the stress responses, and production of many useful metabolites, many useful biomolecules like amino acids. So in today's video, we have discussed the significance of photorespiration. Please, if you find the video useful, hit the like button and share the video with your friends. Thank you.